Okay, so nucleic acids, learning objectives will include knowing what nucleic acids are, knowing how nucleic acids are formed, and describing DNA, the structure of DNA and its properties, knowing also the base pairing rules for DNA, and also the Watson-Crick model of DNA structure, and then comparing and contrasting DNA and RNA. So the outline of our lesson, we're going to look at the introduction and overview of the topic. Then we're also going to look at nucleotide, an overview of nucleotide. We're going to look at the formation of a nucleoside, and then how a nucleotide, a dinucleotide, and a polynucleotide is formed. And then we're going to look at the properties of DNA, and then we'll look at an overview of RNA, and we are going to look at the differences and similarities between DNA and RNA. So when you look at nucleic acids, these are simply large biomolecules which store, transmit, and express genetic information of a cell. These genetic materials are found in all living organisms. They are found in all living organisms. So just like a computer disk contains or stores and transmits information for a computer, or just like a map is to someone who is traveling, storing information about a journey. That's how nucleic acids are. They store, transmit, and express genetic information. Then there are basically two types of nucleic acids. The first one, which is deoxyribonucleic acid, or commonly known as DNA, and the other one is ribonucleic acid, or abbreviated as RNA. So when you look at DNA, DNA simply encodes genetic information that is used to assemble or to make proteins. So the information for protein synthesis is encoded by the DNA. The DNA is a storage material, a genetic storage material. And that information that is stored in DNA is transmitted from one generation to the next generation in the cells. RNA, on the other hand, is normally used as an intermediate in the transfer of information from DNA to proteins. It's a carrier nucleic acid. It's a carrier molecule. Nucleic acids are unique from other macromolecules in that they can serve as templates to produce precise copies of themselves. This means that genetic information is able to be preserved during cell division and during reproduction of organisms. And henceforth, that information is able to be transmitted from one generation to another. So nucleic acids are simply polynucleotides or polymers of nucleotides. They are made up of nucleotides which are arranged in extremely long molecules, henceforth known as polynucleotides. So nucleic acids are polymers of nucleotides. Hence, we need to look at what a nucleotide is, what constitutes a nucleotide. So a nucleotide has three components that we need to know. We have a pentose sugar, which could either be a ribosome sugar, which is found in RNA, or a deoxyribose sugar, which is found in DNA. And then it has a nitrogenous base, which could either be a purine. So among the purines, we have adenine and guanine, or it could be a pyrimidine, which is cytosine, thymine, or uracil. So of importance to note here is that thymine is only found in DNA, while uracil is only found in RNA. And the third component of nucleotide is a phosphate group. And this phosphate group makes DNA to have its characteristic acid. So DNA is acidic because of the phosphate or the numerous phosphate groups that it has. So this is an example of a nucleotide. So you have a phosphate group there. You have a ribose sugar with a difference on the 2' position of the pentose sugar. So in RNA, you have the OH. You have OH in RNA on the 2' position of the pentose sugar. And then in DNA, we have a hydrogen and H in DNA on the 2' position of the pentose sugar. And then we have the nitrogenous base. So the one which is being shown there is a 2-ringed nitrogenous base, which is a purine. And then of note, you find the reaction that takes place between a pentose sugar and a 2-ringed ribose sugar, or rather a 2-ringed nitrogenous base, is between the 1' position of the pentose sugar and the 9th position of the nitrogenous base. So as I mentioned earlier, nucleic acids are long polymers of repeating subunits called nucleotides. So this is just a picture or a slide showing the purines, which are adenine and guanine. And then you have the pyrimidines, which are cytosine, which is found in both DNA and RNA. And then we have thymine, which is only found in DNA, and uracil, which is only found in RNA. So when we look at a nucleoside, how is it formed? How is a nucleotide formed? So we find that a nucleoside is formed by a condensation reaction. So water is given out during the formation of a nucleoside. So a nucleoside is simply formed by a pentose sugar, a reaction between a pentose sugar and a nitrogenous base. A pentose sugar and a nitrogenous base. So for example, we have a deoxyribose sugar reacting with a thymine. So the 1' position, the 1' at the 1', the OH on the 1' position of the deoxyribose sugar will react with the first position of the pyrimidine or the thymine in this case, forming deoxythymidine. And then you can have a deoxyribose sugar reacting with a nitrogenous base of adenine, forming a deoxyadenosine. So this is adenine, which is a purine or a 2-ringed nitrogenous base. So the 1' position, of the deoxyribose sugar will react with the 9th position of the purine, forming a deoxyadenosine, which is a nucleoside. So we are saying that a pentose sugar plus a nitrogenous base will form a nucleoside. If you have a ribose sugar reacting with a nitrogenous base, you have a ribonucleoside. Or if you have a deoxyribose sugar plus a nitrogenous base, you are going to have a deoxyribonucleoside. So for example, in the formation of a ribonucleoside, you can have adenine, for example, reacting with a ribose sugar. It is going to give us a adenosine. Or if we have a uracil reacting with a ribose sugar, it will give us a uridine. If you have guanine reacting with a ribose sugar, it is going to give us a guanosine. So a pentose sugar plus a nitrogenous base will give us a nucleoside. a nucleotide then is formed through a phosphodiester.
A question, sir.
Yes.
Yes, sir. If we tend to look at the absence of uracil in DNA. Now, I was looking at a case where uracil is found in DNA. That is, if we tend to look at germination, the germination of cytosine. Yes. So when the germination of cytosine happens, you discover that uracil will be present in DNA. Now, I don't know whether can that be considered at our level, or is it's also another case.
So when you look at deamination of
Yes,
you're talking about deamination of DNA, isn't it?
Yes. The germination of cytosine, we can look at the conclusion to say uracil cannot be found in DNA. But
yes,
there are cases where when germination takes place of cytosine, when cytosine is germinated, then there shall be a presence of uracil.
Yes.
In those cases, yes. So now I was looking at, now can this also be considered as in our syllabus, or it's also another case where this can be found? Because the conclusion so far, we've concluded that uracil is only found in is not found in DNA but in RNA.
Okay. Yes.
Yes.
So for example, if you have deamination, where you find cytosine being converted into uracil.
Yes, sir.
So what would happen? That happens during a replication. When you look at the DNA replication, where you find that deamination happening, then what would happen is that there is a reaction that takes place that leads to the removal of that uracil from the DNA molecule. So there is an error repairing system. So you have enzymes that, yes, you have an enzyme, uracil DNA glycosylase, which is going to excise or remove the uracil from the DNA molecule.
Okay, so meaning at this instance, we're only going to consider a uracil not being part of DNA unless the germination happens, right?
Yes.
Okay, so then.
Okay.
Yes.
Okay, so when you look at a nucleotide, it simply is formed by the addition of a phosphate group to a nucleoside. So the OH group on carbon 5 of the pentose sugar, the 5' end of the pentose sugar, that OH is going to react with the H of the phosphate in a condensation reaction. In short, water is given out, which will lead to the formation of a phosphodiester or phosphoester bond, leading to a formation of a nucleotide. When for the nucleotide to be formed, there is a reaction between the OH of carbon 3 of the pentose sugar of the first nucleotide and the H of the phosphate on carbon 5 of the second nucleotide. When you look at the dinucleotide, it simply means 2 nucleotides. So for that to be formed, you have the 3' position here of the pentose sugar reacting with the H. So you have the OH of the 3' sugar reacting with the H on the phosphate, forming a phosphodiester bridge between 3' and 5' carbon of the 2 sugars, leading to the formation of what is called a dinucleotide. Okay, so when you add another nucleotide to a dinucleotide, you are going to have a trinucleotide. And then that process continues. More and more nucleotides are added which is going to lead to a formation of a polynucleotide. So this leads to the formation of the nucleic acid. So for example, a reaction between a ribose sugar and adenosine is going to give us a reaction between a pentose sugar and adenine. It's going to give us adenosine. When we react that with one molecule of phosphoric acid, it is going to give us adenosine 5 monophosphate. When we add another nucleotide, it's going to give us adenosine monophosphate when we add another phosphoric acid. When we add a third one, we are going to have adenosine triphosphate, which is commonly known as ATP. So that's how ATP is produced. It's a nucleotide. So now we look at, having looked at how a nucleotide, a polynucleotide is formed, we look at DNA. What is the composition of DNA? What is its property or structure? So DNA has chemical components of which we've alluded to earlier. So it has a polynucleotide chain of DNA which contains a nitrogenous base, a deoxyribose sugar, and a phosphate. The bases that are found in DNA are adenine and guanine, and then we have cytosine and thymine. And then we have a phosphodiester bond that joins the 3' carbon of one sugar to the 5' carbon of the other sugar, as we have already explained. And then the other property is that DNA is a double-stranded molecule. It is a double helix. So each DNA molecule is composed of 2 polynucleotide chains that are joined by hydrogen bonds between the nucleotide bases, between the nitrogenous bases. So we have adenine, which pairs with thymine. So since we have 2 chains or 2 strands, an adenine on one strand will pair with a thymine on the other strand. And then guanine will always pair with cytosine. Henceforth, these strands are described as being complementary, as adenine on one strand is always matched with thymine on the other strand, and guanine is matched by cytosine. Then the chains are antiparallel, which means that one chain runs in the direction of 5' to 3', and the other chain will run in the opposite to the first chain or to the other chain. So it is going to run in the 3' to 5' direction. Then the double-stranded molecule is twisted to form a helix with major and minor grooves. So the base pairs are stackled like a spiral staircase in the interior of the molecule. So when you look at the molecule of DNA, you have the interior part, which is made up of the base pairs, which is generally hydrophobic. So the interior surface of the DNA molecule is hydrophobic. It is made up of the base pairs. And then you have the phosphate groups that are on the outside of the double helix. So I have 2 hydroxyl groups of each phosphate that are involved in forming that phosphodiester bond that we've talked about. Then we have 1 free, which dissociates its proton or hydrogen at physiological pH, which is going to give the molecule a negative charge. So you have the sugar phosphate backbone, and on the molecule or the phosphate group there, you are going to have a proton, which is dissociated, associate leading to the sugar phosphate backbone being negatively charged. So the B form of DNA, which was first described by Watson and Crick, is right-handed and contains 10 base pairs per 10. So you have also other forms of DNA, such as the A form or the Z form. However, of importance for us is the B form of DNA. Then the other point that we need to look at is Chargaff's rule. Chargaff's rule and complementarity of bases. Remember, we've talked about adenine forming hydrogen bonds with thymine. So Chargaff came up with rules which states that the total number of purines should always be equal to the total number of pyrimidines. And the total number of adenine, since adenine is complemented to thymine, the composition of the amount of adenine should always be equal to the amount of thymine. And the amount of guanine should always be equal to the amount of cytosine. So for example, if you have given a percentage of adenine of 30%, let's say adenine is, we are given a question that adenine is 30% in a double-stranded DNA molecule. We are able to find the percentage of the other bases. So since we know that adenine pairs with thymine, simply means that if adenine is 30%, then thymine is also going to be 30%. So from 100%, what we are going to remain with is from 100%, what we are going to remain is 40%, because we have adenine 30%, we have thymine 30%. So 100 minus 60 gives us 40%. And then we know that guanine and cytosine are of equal proportion. That means that they're going to share this percentage, this 40%. So we're going to divide it equally. That will mean that guanine will give us 20% and cytosine also give us 20%. Okay, so that's the importance of Chargaff's rule. Chargaff's rule is also important when we start looking at DNA replication, as it gives us the rules of complementarity or complement bases. So this is showing us the bonding that happens. The bonding that happens. So we have the bonding that happens between. We have the bonding that happens between adenine and thymine, and you have the bonding that happens between guanine and cytosine. So guanine and cytosine will form 3 hydrogen bonds, and adenine and thymine will form 2 hydrogen bonds. So this is simply the Watson-Crick model of DNA that we've touched. So we said that DNA, according to the discoveries, that DNA is made up of major grooves and minor grooves. And then we have 10 bases every 10. So at every 10, you have 10 base pairs. So that's a minor groove and a major groove. And then you have the base pairs in the interior of the molecule. And then you have the phosphate sugar backbone. Phosphate sugar backbone. And then you have the antiparallel nature of DNA. So you have one strand running in the 3' direction and the other strand running in the 5' to 3' direction. On the bases there, you have they also verified the complementarity of bases. So they verified the complementarity of bases. This is the Watson-Crick model of DNA structure. And then we also find the Watson and Crick found that 2 hydrogen bonds form between adenine and thymine, as already mentioned, and 3 bonds form between guanine and cytosine, and also the complementarity of bases. They also found that if each rung of the ladder contained 1 purine and 1 pyrimidine, one width of the helix at each base 10 would be 2.0 nanometer, which is the 2.0 that we can see on the diagram, 2.0 nanometer. Watson and Crick also said or found that there are repeating patterns of 0.34 nanometer and 3.4 nanometer measurements, which are apparent from the model. So each pair of bases is exactly 3.4 nanometer from the adjacent pairs above and below. So each 10 constitutes about 3.4 nanometer of length. So we need to know the characteristic of DNA from there. Then we can look at RNA.
Yes, sir.
Yes.
A question, sir. Yes, when we looked at the sugar phosphate backbone being charged, and we looked at the intra part of the DNA, which is hydrophobic. Now, if you look at these two properties, does it tell us to say DNA, is it hydrophobic or hydrophilic, or it is both? And
Okay.
the other thing is there a biological reason as in to why between adenine and thymine we've got 2 hydrogen bonds and 3 on the other?
Okay.
Yes, sir.
So when you look at DNA, you have the exterior and the interior part.
Yes.
When we are talking about hydrophilicity or hydrophobicity, or hydrophobic and hydrophilic.
Hydrophilic, yes.
Yes. Now, when you look at the DNA molecule, if the DNA molecule is not open, if the DNA molecule is not unwound, what would be reacting with the environment is the exterior part, isn't it? The sugar phosphate backbone. That's one which is going to be reacting with the environment.
Yeah.
Remember, when we are talking about hydrophobic and hydrophilic, we are talking about the reaction of a substance to water, isn't it? Hydro water.
Yes, yes.
Right? Yes. So a normal DNA molecule, I would say it's hydrophilic because it's the exterior, the sugar phosphate backbone that is reacting with the external environment, with the water, and henceforth it becomes hydrophilic.
Okay.
Okay?
Yes.
Is it okay?
Yes, covered on that one.
Yeah, so when we looked at this base pairing,
Yes, the complementarity. Yeah.
yes, we said you've seen the reaction that takes place, isn't it?
Yes.
So when you look at that nitrogen react with that hydrogen, forming this hydrogen bond, isn't it?
Yes.
So
Sir.
first to understand why that will form, why a guanine forms only hydrogen bonds with guanine and cytosine, isn't it? And also have adenine and
Yes.
thymine forming two bonds. For us to consider this structure. So the way my understanding is from the experiment that Watson and Crick performed, that led to their conclusion of the bonding that happens of biological significance between this bond pairing. Is that when you look at the molecule of DNA itself, according to their research, it was found that for this molecule of DNA, for this molecule of DNA to be formed as it is in its complementarity, as we have looked, it has to be. You look at the functional groups. So you have remember you have the double bond oxygen there and the hydrogen there. So my understanding is simply the reaction between these atoms in terms of the functional groups. Remember,
yes,
bonding in a chemical substance is really dependent on the distribution of the electrons, right?
yes, yes.
Yes. Now, when you look at that hydrogen there on formation of adenine and thymine there, between the nitrogen, you can see the picture there down, right?
Yes, yes.
Yes, you have the hydrogen, the unpaired hydrogen, the oxygen, isn't it? There's a double bond between nitrogen and nitrogen and the carbon attached to the hydrogen, isn't it?
To this hydrogen.
Yes, so which means that the hydrogen has formed a maximum number of, or it has shared a maximum number of electrons, or it has shared, yes, with a